In Lab 3, we're going to be looking at how to obtain pure cultures from a mixed population. This is another lab where you have several videos to watch, one on aseptic technique, streaking for isolation. You want to make sure you review that we had in Lab 2, because streaking to get isolated colonies is critical to today's lab. And then a second video on how to obtain pure cultures from isolated colonies. There's also a brief soft chalk lesson on the 3 agars we're using today: tryptase soy agar, Columbia CNA agar, and MacConkey agar. And of course, remember your performance objectives at the end of the lab tells you what you'll need to know for the lab quiz covering this lab. This will be the last time I'll go through the performance objectives. After today, I'll just assume you're looking at them. But after today's lab, when given a mixture of a gram-positive and a gram-negative bacterium and plates of Columbia CNA, MacConkey, and tryptase soy agar, describe the steps you would take to eventually obtain a pure culture of each organism. You should be able to define selective medium, differential medium, enrichment medium, and combination selective differential medium. You should be able to state the usefulness of Columbia CNA agar and MacConkey agar, and describe how each of the following would appear when grown on MacConkey agar: Escherichia coli, Klebsiella aerogenes, formerly known as Enterobacter aerogenes, and Pseudomonas aeruginosa. As far as core lab points, you should be able to streak a plate for isolation, obtain isolated colonies from a mixture of microorganisms, and then be able to pick off isolated colonies from the isolation plate and aseptically transfer those to a sterile medium to obtain pure cultures. And practical wise, when given a plate of Columbia CNA agar or MacConkey agar showing discrete colonies, correctly interpret the results. And of course, remember, as always, there's a self-quiz with answers at the end. So let's get into today's lab. Now, as we mentioned in Lab 2, in nature, microorganisms exist as mixed populations. But if we want to study organisms in the lab, we typically need them in the form of a pure culture. And as we defined in Lab 2, a pure culture is one in which all organisms are descendants of the same organism. So first thing we need to realize is that there's 2 major steps in obtaining pure cultures from a mixed population, and it will take us a total of 3 labs to get those pure cultures. The first thing we have to do is take the mixture and dilute it until various individual microorganisms become separated far enough apart on the agar surface that after incubation we get colonies isolated from the colonies of other microorganisms. So what we're doing in step 1 is trying to get good isolation plates. And an isolation plate is simply one that has at least some isolated colonies that can be picked off in order to obtain a pure culture. And that brings us to the second step. Once we have isolated colonies, then we can aseptically pick off the isolated colonies from the isolation plate and transfer those to a new sterile medium. And after incubation, then we will have a pure culture. Because remember, in the isolation plate, we're looking for single isolated colonies. And we said a colony was a visible mass of microorganisms on an agar surface that came from one organism or one arrangement of that organism. So if we pick off part of a single colony, which came from one organism, and transfer that to a new sterile medium in step 2, we will have a pure culture, one in which all cells are descendants of the same cell. So there are several ways we can get isolated colonies, and the most common technique, and the one we're doing today, is what we learned in Lab 2, the streak plate method of isolation. So what we're doing here, of course, is streaking the loop across the agar surface, so more and more bacteria are rubbed off and isolated, so we get separate organisms somewhere on the agar that after incubation give us isolated colonies. Now let's take a look at how we go about doing that before we get into the other methods. So let's go back up here where we pick off isolated colonies. If we look at figure 1, we see a streak plate on tryptocase soy agar with one of the mixtures we're using today, a mixture of Micrococcus luteus and Escherichia coli. And notice on this plate, we have single isolated colonies of Micrococcus luteus, as shown by the yellow water-insoluble pigment that Micrococcus luteus produces, and single gray colonies, more regular looking, that is the Escherichia coli. So if we're to pick off part of that Micrococcus luteus colony from the isolation plate and then streak that on a new plate of tryptocase soy agar, we would have a pure culture of Micrococcus luteus. If we then picked off a single colony of Escherichia coli, streak that on a second plate of tryptocase soy agar, that would give us a pure culture of Escherichia coli. So the idea, as we see in figure 3, is that if we pick off part of one colony from one of the organisms in your mixture and streak that for isolation on a new plate of TSA, that would be a pure culture of this organism, because this culture came from one colony, which came from one organism or arrangement of that organism. Then if we were to pick off a colony of the other bacterium, streak that on a different plate of TSA, that would give us a pure culture of that organism. So that's our streak plate method of isolation. And again, here's another plate where we can see single colonies of Micrococcus luteus with the yellow water-insoluble pigment and single colonies of Escherichia coli. And again, that would be a good isolation plate because there's single colonies we can pick off and transfer to new medium to obtain pure cultures. Now, a second technique we can use to get isolated colonies is called the pour plate method. In this case, the bacteria are mixed with melted agar that's cool enough that it won't harm the bacteria. And we mix that up so the bacteria become evenly distributed throughout the liquid agar. We pour the melted agar on empty plates and allow it to solidify. And after incubation, there should be discrete colonies found growing both on the surface and in the agar that we could pick off. We're not going to do the pour plate method. That's a little more cumbersome, and the streak plate method works very good. A third method is the spin plate method, which we're going to do in lab 4, enumeration of microorganisms. Now, in this case, we first dilute the bacteria in tubes of sterile water or saline or broth. And once we've diluted the bacteria, small samples of the diluted bacteria can be pipetted onto the surface of an agar plate. Then we use a sterile bent glass rod and a turntable to spread that mixture of bacteria over the entire agar surface. And that allows us to see eventually isolated colonies. So figure 4 shows you the method we'll be using in our next lab, lab 4, where we first diluted the bacteria in a liquid, then we pipetted a small sample on a Petri plate. We take a bent glass rod that we've sterilized by dipping in alcohol and then catching the alcohol on the rod on fire and letting it burn off. Then we lay the glass rod flat on the agar surface and spin the plate using this turntable, and that spreads the bacteria evenly over the entire surface. And so after incubation, we should see some single isolated colonies like we see in figure 5. Now we're going to use this in lab 4 in order to get single colonies that we can count. And we'll use that as a way to determine the amount of bacteria in a sample. We also need to talk about the use of specialized media. So far, we've only used tryptocase soy agar, kind of an all-purpose growth medium that pretty much grows anything. But most of the time, we want to try to isolate specific groups of organisms or help identify those organisms. And for that, we use special purpose media. And there's hundreds of special purpose media that we can purchase for different microbiological techniques, but they fall into 4 major groups. The first group is a selective medium. Now, a selective medium has agents added that inhibit the growth of one group of organisms while permitting the growth of another. An example of that we're using today is Columbia CNA agar. And Columbia CNA agar contains the antibiotics colistin and nalidixic acid, which is what the CNA stands for. And these antibiotics inhibit the growth of gram-negative bacteria, but they don't affect the growth of gram-positives. So we say that Columbia CNA agar is selective for gram-positive organisms. That is, gram-positive bacteria will grow and gram-negative bacteria would be inhibited. But an important point to remember here is that even if you only see the gram-positive bacteria growing on Columbia CNA agar that was streaked with a mixture of a gram-positive and a gram-negative, that's not a pure culture. There's still two bacteria growing on the plate. The gram-negative bacteria are inhibited, not killed. They're growing more slowly. They're still alive on the agar surface. They just haven't grown in sufficient quantity that we can see visible colonies. So we still have to pick single colonies off of the Columbia CNA agar plate and streak those on a new agar plate before we get a pure culture. That's step 2 of our procedure for getting pure cultures. A second category of medium is a differential medium. Now, a differential medium has additives that cause an observable color change in the medium when a particular chemical reaction occurs. So we identify bacteria largely by their biochemical characteristics. Because each bacterium has unique DNA, it makes unique enzymes and carries out some unique biochemical reactions. So a differential medium tells us whether or not a certain microorganism can carry out a specific biochemical reaction as part of its normal metabolism that we can use to help identify that bacterium. We're not going to use a strictly differential medium today, but we'll use many of these during the semester to help us identify different microorganisms. Our third category is called an enrichment medium. Now, this has additives that enhance the growth of certain organisms. So this becomes useful when the organism we're trying to isolate is present in relatively small amounts compared to other organisms growing in the mixture. In this case, we add enrichment additives that enhance the growth of the one we're trying to isolate so more of them grow so we have a better chance of getting isolated colonies. We're not going to use any strictly enrichment medium in our labs this semester, but that is a category that's used sometimes for more fastidious or hard to grow organisms or organisms that are often present in relatively small in number compared to other microorganisms present at that location. And our final category of medium is a combination of 1 and 2 up above, a combination selective and differential medium. So the selective part permits the growth of one group of organisms while inhibiting another, but the differential part also allows us to differentiate those organisms that grow based on whether they carry out a particular biochemical reaction. And a nice example of that we're using today, and again in Lab 12, and for some of you on your final lab project, is MacConkey agar. Now this is an agar that normally has, before you inoculate it, kind of a reddish color as we see in figure 6. And we use this as a selective medium for isolating non-fastidious gram-negative rods. We particularly use this to identify members of the bacterial family Enterobacteria, as well as another genus of gram-negative bacillus, Pseudomonas. And we'll take up the family Enterobacteria as well as the genus Pseudomonas in labs 12 and 13. So on this medium, gram-negative bacteria grow, gram-positives are inhibited. But it's also a differential medium in that it differentiates lactose fermenting from lactose non-fermenting gram-negative bacilli. Now, the way this medium works, MacConkey agar contains the dye crystal violet as well as bile salts. And the combination of crystal violet and bile salts inhibits most gram-positive bacteria, but doesn't affect the growth of gram-negatives. So we say MacConkey agar is selective for gram-negative bacteria. Gram-negative bacteria grow, gram-positives are inhibited. And then if the gram-negative bacterium that grows is able to ferment the sugar lactose, we can detect that in the medium. Because the end product of fermentation, in this case fermentation of lactose, is acid end products. And as the acid end products accumulate in the agar, that lowers the pH. And that causes the neutral red in the agar to turn red in color once the pH drops below 6.8. And as the pH drops, the neutral red is absorbed by the bacteria, causing the colonies to appear bright pink to red. So if the bacteria grow on MacConkey agar and produce pink to red colonies, we know it's gram-negative because it's growing on MacConkey agar, and that it's fermenting lactose because we see the pink to red colonies, indicating lactose fermentation. But we can also differentiate strong fermentation of lactose from weak fermentation of lactose on this medium. Strong fermentation of lactose produces high levels of acid produced by the bacteria. And of course, as we just said, the acid accumulation causes the colonies as well as the confluent growth. The confluent growth is where you have solid masses of bacteria growing together to appear bright pink to red. But if there's strong fermentation with high levels of acid production, resulting acid at high enough concentration causes the bile salts in the medium to precipitate out of solution. And that causes a bright pink precipitate or cloudiness to appear in the agar surrounding the colonies. And we can see this in figure 7. So this is Escherichia coli growing on MacConkey agar, and Escherichia coli strongly ferments lactose. We can tell it ferments lactose because the colonies themselves, which we see here, as well as the confluent growth up here, appears bright pink to red. But we can tell it's strong fermentation because here, where this arrow is, we see the precipitation of the bile salts in the agar, causing a bright pink precipitate or cloudiness to appear in the agar surrounding the growth. So here we see the bright pink precipitate. We see it down here. We see it around some of the individual colonies. And that tells us there's strong fermentation of lactose. If there's weak fermentation of lactose, then again, the acid causes the colonies and the confluent growth to appear pink to red, but there's not enough acid to precipitate out the bile salts, so we see no pink precipitate surrounding the growth, and we see that in figure 8. So again, we see bright pink to red colonies, indicating that there is fermentation of lactose, but notice there's no bright pink precipitate out here in the agar surrounding the growth of the colonies. And so since we don't see that bright pink precipitate or cloudiness, that tells us it's weak fermentation rather than long fermentation. So for example, Klebsiella aerogenes that we're using today, which was formerly known as Enterobacter aerogenes, shows weak fermentation on MacConkey agar. And the third possibility is that the bacteria do not ferment lactose. So if there's no fermentation of lactose, we don't see any pink or red colonies. The colonies appear pretty much colorless, and the agar surrounding the bacteria remains relatively transparent, as we see in figure 9. This is Pseudomonas aeruginosa growing on MacConkey agar, and Pseudomonas does not ferment lactose. So notice there's no pink to red colonies anywhere. There's no bright pink precipitate, and that shows us that lactose was not fermented. But again, remember that if we only see the gram-negative growing on a plate of MacConkey agar that was streaked with a mixture of gram-positive and gram-negative bacteria, that's still not a pure culture. The gram-positive bacteria on the agar are inhibited, not killed. They're still alive on the agar surface. They just haven't grown sufficiently to give us visible colonies. So we still have to pick colonies off of the MacConkey agar and streak those on a new agar plate before we get obtain our pure culture. So the MacConkey agar is our isolation plate, giving us single colonies, but until we pick off single colonies from the MacConkey agar and streak those on a new agar plate, we don't have a pure culture. So there are 3 agars we're going to use today in lab, and you had a short soft chalk lesson on those, as well as mentioned up above. Tryptocase soy agar is kind of an all-purpose growth medium. It grows both gram-positives and gram-negatives. But the problem with tryptocase soy agar as an isolation medium is that if you have a mixture of 2 bacteria, you may not see single colonies of both, although both are able to grow on this medium. And the reason for that, excuse me, is that often one bacterium in the mixture overgrows the other. It is much more abundant. So by the time you get the more abundant bacteria spread out far enough to get single isolated colonies, you've lost all the bacteria on the loop of the one that's in the minority. So it is possible on trypticase soy agar, you're only going to see single colonies of one of the two bacteria in the mixture because one's overgrowing the other, unless they're fairly equal in number to begin with. Now our second agar, Columbia CNA agar, remember, is selected for gram-positive bacteria. So the gram-positive bacterium in your mixture will grow, the gram-negative will be inhibited. And the MacConkey agar is selected for gram-negative bacteria. The gram-negatives will grow, the gram-positives will be inhibited. And we'll see how we inoculate those media shortly. But let's first talk about the organisms we're going to use today. Now, each pair is going to pick a mixture of bacteria. We have several different mixtures, but every mixture has 1 gram-positive bacterium and 1 gram-negative bacterium. So your job between now and lab 5 is to obtain a pure culture of the gram-positive bacterium you have in the mixture, as well as the gram-negative bacterium you have in the mixture, and determine which gram-positive bacterium and which gram-negative bacterium you had in that mixture. And these will be your pure cultures for lab 5. We're going to stain these 2 bacteria in lab 5 and see if they look pure. So to start out today, your mixture, as we said, has 1 gram-positive bacterium and 1 gram-negative bacterium growing in it. Now, there's 2 possible gram-positive bacteria today. You will have one of these 2 in your mixture, either Micrococcus luteus or Staphylococcus epidermidis. To help us out when we stain these in lab 5, Micrococcus luteus is a gram-positive coccus with a tetrad or sarcina arrangement. Staphylococcus epidermidis is a gram-positive coccus with a staphylococcus arrangement. But what we're going to be looking for are colonies of these bacteria on the plate, and we're going to identify whether your gram-positive is Micrococcus luteus or Staphylococcus epidermidis by pigment production. Because as we learned in lab 2, Micrococcus luteus produces circular convex colonies that produce a yellow water insoluble pigment. And you'll be able to see that on the trypticase soy agar if you have single isolated colonies of Micrococcus luteus on trypticase soy agar. And you'll also be able to see this on the Columbia CNA agar, where only the gram-positive will grow. So again, if we see that the organism is producing a yellow water insoluble pigment, then you know your gram-positive is Micrococcus luteus. Whereas Staphylococcus epidermidis also produces circular convex colonies, but does not produce pigment. So these colonies will appear white on the trypticase soy agar if you have single colonies of that growing on the trypticase soy agar, and will appear white on the Columbia CNA agar as well. So if we don't see the yellow pigment on the Micrococcus luteus, then we know that your unknown is Staphylococcus epidermidis, a non-pigmented bacterium. Just as an example, here's a plate of Micrococcus luteus growing on TSA, where we see the circular convex colony of Micrococcus luteus producing the yellow water insoluble pigment. The Escherichia coli and any of the gram-negatives we're using today will actually produce bigger colonies on the trypticase soy agar, because E. coli tends to grow better than the 2 gram-positives we're using today. But the E. coli produces no pigment, so the colonies appear gray, and they have a more irregular edge and they're larger. In the case of Staphylococcus epidermidis, if it's growing on TSA, notice that this again has circular convex colonies, but no pigment, it's white. So that would be the Staph epidermidis, and the larger, more irregular edged, non-pigmented gray colony, in this case, is the Escherichia coli. Now, in addition to one of these 2 gram-positives, Micrococcus luteus or Staphylococcus epidermidis, your mixture will have one of 3 possible gram-negative bacteria, either Escherichia coli, And all three of these are gram-negative bacilli when we look at them in lab 5. But Escherichia coli produces irregular raised non-pigmented colonies on trypticase soy agar. Klebsiella aerogenes, formerly known as Enterobacter aerogenes, produces very similar colonies to the Escherichia coli. There are irregular raised non-pigmented colonies as well. And we can't really tell the Escherichia coli from the Klebsiella aerogenes on trypticase soy agar because their colonies look similar. But remember, when we get to MacConkey agar, we'll be able to tell them apart because Escherichia coli strongly ferments lactose and Klebsiella aerogenes weakly ferments lactose. And we'll mention that again when we get to the MacConkey agar down below. Now, the third possible gram-negative bacteria in your mixture is Pseudomonas aeruginosa, another gram-negative bacillus. This too has irregular raised colonies, but remember we learned in lab 2 that Pseudomonas aeruginosa produces a green to blue water-soluble pigment. And so if we see that a green water-soluble pigment is being produced, we know then that your gram-negative is Pseudomonas aeruginosa. So during the next couple of labs, we're going to try to obtain pure cultures of each of the 2 organisms in your mixture and determine which 2 you have, which gram-positive bacterium you have, and which gram-negative bacterium you have. So today we're going to try to separate the bacteria to obtain good isolated colonies on what we call isolation plates. Next lab, after these plates incubate, we're going to identify which 2 bacteria you have in your mixture, then pick off a single colony of each of the 2 bacteria you identified in your mixture, streak those on new plates of trypticase soy agar in order to get pure cultures. And then the following lab, after those incubate, we're going to be staining those and looking at them under the microscope and see if they are pure. So the procedure today, on all 3 plates, of course, you want to divide the plates up like we normally do to get isolated colonies to guide your streaking. So divide it into 3 sectors. Then you're going to take the mixture that you pick and streak it for isolation on trypticase soy agar. Now remember, that grows both gram-positive and gram-negative. And as I mentioned previously, trypticase soy agar is not normally used as an isolation medium. So keep in mind, if you're answering a quiz question on obtaining pure cultures, we wouldn't normally use trypticase soy agar to get isolated colonies. We would use that to grow the bacteria on after we pick off colonies. But we'll streak it on the trypticase soy agar anyway and see if you do get single isolated colonies of both. And that will give you more practice on streaking Petri plates for isolation. So there we're just using mechanical methods to try to separate the bacteria rather than using chemicals that inhibit one group or another group. So streak your mixture on a plate of trypticase soy agar, and let's review our procedure. Now again, in the videos for today's lab, I've repeated the one on how to get isolated colonies using the streak plate method. So make sure you review that video because it's essential that you streak the plate properly today in order to obtain single isolated colonies. So remember, when we streak the plates today, as we see in figure 4, you'd first streak your mixture over sector 1. Now remember, you're trying to spread the bacteria out over the entire agar surface in sector 1. So you want to go all the way to the edge of the plate, back and forth, keeping the lines close together so it keeps spreading them out and spreading them out and spreading them out. Once you finish streaking sector 1, remember, you're going to flame the loop by putting it in your bacterial incinerator to kill any bacteria on the loop, and then let the loop cool. Then we're going to rotate the plate counterclockwise so that sector 1 is now sitting at 9 o'clock, as we see in the next step. You're going to take your sterile inoculating loop and drag that through the bacteria you've already streaked on sector 1 to pick up some of the bacteria here and spread those out over sector 2. Now, it's very important when you start streaking the plate from sector 1 to sector 2 that you go to the very edge of the plate and draw it through the whole mixture here. Don't start your streaking here because you may not pick up any bacteria to spread out. You want to make sure you do pick up some bacteria on your loop, so you want to start here to the far left of the plate, drag it through no more than 2 times because we don't want to pick up too many bacteria, and then streak that out all over sector 2 without dragging it through. Then at this point, you would put the loop in your Bacto incinerator to flame it and sterilize it and let it cool. Then we'll rotate the plate counterclockwise again, so sector 2 is sitting at 9 o'clock as we see in step 3. So now we take our sterile inoculating loop, starting at the far left of the plate to make sure we pick up bacteria. We drag it through the bacteria that we've spread out on sector 2, drag it through no more than 2 times, and then spread that out carefully over sector 3, being careful not to drag it through more than 2 times, and making sure we don't touch sector 1 where they're most concentrated, because it's in sector 3 that we'll most likely see single isolated colonies. So again, you want to use your best streaking method. Now you're streaking 3 Petri plates today per pair, so you can divide it up where one of you does 1 plate and the other person can do 2 of them. After you streak your mixture on a plate of trypticase soy agar, you're going to take the same mixture and streak it for isolation on a plate of Columbia CNA agar, which remember is selected for gram-positive bacteria. So on the Columbia CNA agar, only the gram-positive bacteria in your mixture will grow well. The gram-negative will be inhibited. And if you don't see single isolated colonies from the trypticase soy agar, you will definitely be able to get them on the Columbia CNA agar because only the gram-positive will be growing. And again, if you have Micrococcus luteus on the Columbia CNA agar, we're going to see the yellow water-insoluble pigment. So if we pick off a single colony, say from the Columbia CNA agar next time, and streak that on a new plate of agar, that will give us a pure culture of Micrococcus luteus. On the other hand, if you have Staphylococcus epidermidis, when you grow that on Columbia CNA agar, it will have no pigment. It'll appear white. So if we pick off a single colony of Staphylococcus epidermidis from the Columbia CNA agar, streak that on a new agar plate, that would give us a pure culture of Staphylococcus epidermidis. So we use pigment to determine which of the 2 gram-positives you have. Then finally, you're going to streak the same mixture for isolation on a plate of MacConkey agar, but this is selected for gram-negative bacteria and differentiates gram-negative bacilli according to whether or not they ferment lactose, and if they do, how strongly they ferment lactose. So it's on the MacConkey agar that you'll be able to definitely determine which gram-negative bacteria you have. As we discussed under MacConkey agar up above, Escherichia coli shows strong fermentation of lactose. And we can tell it's strong fermentation because the colonies have turned bright pink to red, indicating fermentation of lactose. What tells us it's strong fermentation is this pink precipitate in the agar. Bright pink opaque precipitate or cloudiness. So if your MacConkey agar looks like this next time, showing strong fermentation, you know your gram-negative is Escherichia coli. If we're to pick off a single colony of the Escherichia coli, streak that on a new plate of TSA, the following lab we would have a pure culture of Escherichia coli. If on the other hand, your gram-negative is Klebsiella aerogenes, Klebsiella aerogenes weakly ferments lactose. So now we'll see the pink to red colonies indicating lactose fermentation, but because there is no bright pink cloudiness or precipitate in the agar, that shows us that it was weak fermentation rather than strong fermentation. So if we see weak fermentation, then your gram-negative today is Klebsiella aerogenes. If we pick off a single colony from the MacConkey agar here and streak it on a new plate of TSA, that will give us a pure culture of Klebsiella aerogenes. And finally, if your gram-negative is Pseudomonas aeruginosa, on the MacConkey agar, Pseudomonas does not ferment lactose. So there's no pink or red colonies anywhere on the plate. The colonies are relatively colorless, and the agar itself is relatively colorless and rather transparent looking. So if we pick off a single colony from this plate, in this case Pseudomonas aeruginosa, because it shows no fermentation of lactose, and streak that on a new plate of TSA, that would give you a pure culture of Pseudomonas aeruginosa if that's the gram negative you have. So once you streak your TSA plate, your Columbia CNA agar plate, and your MacConkey plate for isolation, then you're going to take those 3 plates and incubate them upside down in your Petri plate holder in 37-degree incubator on the shelf corresponding to your lab section. Then next time you're going to look at the results of these 3 agars and write your observations and your conclusions for each of the 3 agars. Then using any of the 3 plates on which the organism is growing, you're going to aseptically pick off a single colony of each of the 2 bacteria in your original mixture and aseptically transfer those to separate plates of trypticase soy agar, streaking for isolation again in order to get pure cultures. So we're always going to use a streak plate method of isolation when we streak a Petri plate unless otherwise mentioned. So again, you just want to find out which plate of the 3 has a good single colony of the gram positive in your mixture that you identified. Most likely it'll be easiest to get that gram positive off of the Columbia CNA agar, which grows only the gram positive. But you might see single colonies of your gram positive on the trypticase soy agar. And again, we're using pigment to determine which gram positive you have. Micrococcus luteus with a yellow water-insoluble pigment, or Staphylococcus epidermidis that has no pigment and produces white colonies. So you want to pick off a single colony of whichever gram positive you identified from any agar where you see isolated colonies of that specific organism and streak that on a plate of TSA and incubate it. Then you want to pick off a single colony of whichever gram negative you had. Most likely it'll be easiest to get it off the MacConkey agar where only the gram negatives grow. But you might see some good isolated colonies on the trypticase soy agar, and you know which one you have based on the MacConkey agar. Pick off that colony, streak on a plate of TSA to get a pure culture of your gram negative. Now, when you pick off the single colonies next time from the TSA or the Columbia CNA or the MacConkey, try to remove the top part of the colony without scraping the agar surface itself if you can. That way you won't pick up any of the inhibited bacteria that might be living under the colony. Now make sure when you pick off colonies next time and streak them onto TSA to get pure cultures the following lab, that you write the name of the bacterium that you identified, the genus and the species that you're growing on the new plates of TSA for lab 5. So again, don't write that it's gram positive or gram negative or the agar it came from. You need to know the name of the organism you're growing and staining in lab 5. So make sure you label it with the genus and species of the bacterium you identified. One plate of TSA will have the gram negative you identified. The other plate will have the gram positive you identified. And take these 2 plates of TSA and incubate them in your Petri plate holder in the 37-degree incubator until the next lab period. And then these will be your pure cultures that will be staining in lab 5 on direct and indirect staining. So that's the technique we use to obtain pure cultures from a mixed population.